Hi, and welcome to this video where we're going to be running through the November 2020 Biological Processes paper from the OCR exam board. Now, in this video, you can do it in two ways. Either if you're doing this as a run-through video, you can kind of follow along, or if you're using this to mark and annotate a paper that you've already done, then you need to do this paper first. So in this video, we're going to be looking at the content you need to know, but also the skills and the ways of addressing, ways of answering exam questions, and also looking at some examples of people work, the mark scheme, and some common pitfalls or traps that you could fall into. So the first thing I'm going to do is I'm going to show you the mark scheme for the multiple choice section, because this video is going to be fairly long. So first you can just mark your multiple choice section if you've already done it, and then you'll be able to skip through to the questions on this part which you didn't get. Okay, so pause the video here, mark your multiple choice, and if you don't know why you've gone wrong, we're going to go through those questions now. Okay, let's approach all the multiple choice questions, and see how you get the answers. Okay, question one. Which of the following statements A to D about differential staining is not true? Differential staining can distinguish between different organelles. That is true. You can stain different organelles with different dyes and therefore get different colours, so that's true. Differential staining can distinguish between types of cell. That's also true. Differential staining can distinguish between types of organism. That is also true. You could have very small organisms that you're looking at. Differential staining is a common feature of electron microscopy. That is not a common feature of electron microscopy. So the answer is D. Okay, question two. This is a tricky one about microscopy. So first of all, this one, light microscopes with an eyepiece lens of ×10 and an objective lens of ×50, the magnification, you multiply them together. So that would be ×500. So this one is incorrect. This one, B, scanning electron microscopes and laser scanning can both produce three-dimensional images. Now, is that correct? We can kind of put a little question mark on. It could be a possible. Well, let's keep going. The maximum resolution of a scanning electron microscope is lower than that of light microscope. No, that's not true. Electron microscopes always have a higher resolution than light, so that's wrong. D, transmission electron microscopes and laser scanning confocal can both produce three-dimensional images. That's definitely wrong because transmission is wrong. Now, the reason I question mark this one is because laser scanning confocal microscopes, I'm not even really that sure what that is. Confocal microscopes tend to take an optical slice through a thing, through a specimen, sorry. But if you build up many, many layers, I guess you could probably make a 3D image. So it's definitely the least wrong, so we're going to put it, and that's what the exam board says. 3. Okay, here is a slice of plant tissue showing the stem. Okay, and you need to basically remember this, what's what. So we could just sort of start by labeling. That's the cambium right there. G, this is the xylem. You can see these big empty cells here. And F is the phloem. So therefore, okay, this is right. Function of S is transport to stimulus, that's right. And transport of water, that's right. So the answer is A. This is really just a knowledge question here. But you do need to recognize this kind of picture and be able to label the parts. Moving on. 4. Which of the following statements, A to D, does not correctly describe the structure or formation of plant vascular tissues? Okay, companion cells are linked to xylem cells by plasmodesmata. That's not correct. Okay, the xylem, they're linked to phloem cells, so that's definitely incorrect. So I think it's that one, isn't it? Let's just double check that. Yeah, that's right. So let's always check each one and see which one's true and which one's false. Mature sieve tube elements do not contain nuclei. That's true. C, phloem and xylem are formed by differentiation of vascular merostems. That is true. Xylem vessels have non-lignified pits to allow movement of water in and out, sort of out of the xylem. That's true. So yeah, it is definitely A. Okay. Rest assured. Moving on to 5. Large multicellular animals need a transport system for oxygen and carbon dioxide. Yep. Large multicellular plants do not need a transport system for oxygen and carbon dioxide. Yep. Which explains these observations? Well, before we even look at the observations, it's to do with kind of levels of activity. So let's just think. Large plants have a low surface area to volume ratio. Now that doesn't explain it. That's a reason why they do need a transport system. Plant cells have a low metabolic rate. That does explain it. Plants generate ATP during photosynthesis, so they do not need to respire. Now that's definitely not true. Plants respire and do photosynthesis. Plants generate oxygen during photosynthesis. Well, that's true, but it doesn't explain it. So that's nothing to do with need for exchange or the need for a circulatory system. The answer is 5B. Plants don't run, they don't move about, they're not as metabolically active as, let's say, a brain cell or a muscle cell, so therefore they don't need lungs. 6. Which of the following statements, A to D, describes the movement of water across plant roots? Okay, so which is correct, basically? The Casparian strip blocks movement by the symplast pathway. Apoplast, vacuolar, symplast. Okay, so no, it doesn't. Okay, the Casparian strip, the symplast pathway, this one, is through the cytoplasm. Okay, so the Casparian strip doesn't block that. It blocks the apoplast pathway, which is through the cell wall. Symplast pathway requires water to cross partially permeable membranes. That is true, because they have to go across the cell membrane into the cytoplasm. Water, although there are plasmodesma, I'm going to come back to that and keep going, just double check. C, water moves from the soil to the root hair cells up a water potential gradient. No, it's down a water potential gradient, so that's false. Water moves through the leaves only by the symplast pathway. No, that's not true. So yeah, B is true. The reason I hesitated is because there are sometimes plasmodesmata between adjacent cells, so it doesn't necessarily always have to go across the membrane to get from cell to cell, but I guess it will at some point. 7. Which of the following statements about water transport in plants is or are correct? Okay, transpiration happens as a consequence of the need for gas exchange. Whoosh, these are hard. I'm going to say yes, because without, the leaves have stomata, and the stomata is because of gas exchange, and without stomata, you wouldn't get transpiration. So that's my thoughts. 2, there are cohesive forces between water molecules because they form hydrogen bonds with one another. Yes, true. 3, water is drawn up the stem. This is, gosh, I haven't done one of these for a while. I've forgotten how horrible these papers are. So this is wrong because adhesion is the stickiness of water molecules to the actual sides of the xylem vessel. Cohesion is together. So between water molecules and adhesive, that's like a disconnect. Adhesion between the water molecules, no, so that's not right. So the answer is only 1 and 2. My word, picky. You've got to be really on it with your vocabulary. 8, I like this question. It's a testing one, but I like it. So first of all, gas exchange in bony fish. Hopefully you remember that this is a countercurrent exchange mechanism, and hopefully you will just remember that it's one of these graphs, A, B, or C. You might just remember, A, B, or D even. You might remember this kind of general shape of it. Now the first thing you need to look at is which is countercurrent. So you need to look at these parts down here, okay? Is this countercurrent? Water going one way, blood going the other way? Yes, it is. Is this countercurrent? Water going one way, blood going the other way? Yes, it is. Is this countercurrent? No. Look, the water and blood are going the same direction. Okay, so we've got it down to 2. How can we figure out the last one? Well, now the only difference, first of all, it's to spot the difference. What is the difference between these two? Well, we've got blood and water. Here we've got water and blood. So which line should be the highest and which should be underneath? Which thing has the higher oxygen saturation? Well, it's the water. The water has got high oxygen, and it's always going to be diffusing from the water into the fish's blood. So we want a situation where the water is above. So that's this one. So the answer is therefore B. 8 is B. Love that question. Moving on. Okay, so 9 and 10 both refer to this. Okay, so I think first thing is just to read it and understand what on earth is going on. Okay, so I'm getting our highlighters out. Extract of the bark of the Indian fig tree has antioxidant properties. Ficus racemosa could protect against damage to non-cancerous cells during radiotherapy. Okay, so protects against damage to non-cancerous cells during radiotherapy, this antioxidant. Cultures of non-cancerous cells were exposed to increasing doses of radiation, and the percentage of damaged cells was measured. The experiments performed multiple times, either with or without the bark extract. I got you. Okay, the graph shows the results for the two sets of data. Each data point shows the mean plus a standard deviation, so it's the spread of the data. Now, the first thing I'd probably do is kind of just get an estimate. Look at this. Okay, so without the extract, this is kind of the line of best fit. And with the extract, this is the kind of line of best fit. It does seem like there's a big difference there. So let's have a look at the questions. A student used Spearman's rank correlation coefficient to test for the correlation between radiation dose and cell damage. Well, looking at both of these things, it looks pretty clear that there is a strong correlation in both cases. But let's read the question. Spearman's rank correlation coefficient for cells without extract were between 0 and +1. That means that we have a correlation. Yes, there's clearly a correlation. I think that's right. Spearman's rank correlation coefficient RS for cells with extract were between 0 and -1. No. So this is basically just means between 0 and +1 means positive correlation. 0 and -1 means a negative correlation. So it's definitely a positive correlation. This again says negative correlation. No, that's definitely wrong. And no correlation, basically, if we had no correlation, the points would just be all over the place or kind of scattered around. So there'd be no pattern, basically. So the answer is A. Again, this is just a horrible way of asking you what type of correlation is there. And the answer is positive. 10. Which of the following statements, A to D, about the graph is correct? The graph for the with extract group is more variable than the without extract. Okay, which is more variable? Yeah, the with extract is more variable. Look at the size of these bars here. Like, for example, this bar was pretty big compared to this bar, it's pretty small. Okay, or smaller. So definitely with extract has higher variation. So I guess we just go straight to A. Is that right? Yeah, it is right, but let's just check the other ones. The error bar shows the range of data. That's actually not true. If you read the question carefully, it was the standard deviation of the data. C, the error bars show whether the difference between two sets of data is significant. That's saying too much. It's not really that. They kind of give you a bit of a clue, but it's not really that. The chi-squared test can be used to determine whether there is a significant difference between the two sets of data. That's also wrong, because chi-squared is about looking at the difference between the observed and the expected data. So you expect one thing, but then you observe something else. And do you know if it's totally different or is it close enough, basically? All right, moving on. Okay, another kind of double question. 11 and 12 refer to the investigation described below. We've got two marks to get here, so let's make sure we understand the experiment. It's a respirometer. We've got some germinating seeds in here. We've got the soda lime absorbing carbon dioxide, so any carbon dioxide produced in respiration is going to be absorbed. Seeds are respiring, they're going to be producing CO2. Now, remember, seeds are not photosynthesizing. Yes, they're plant seeds, but there's no light, probably, and they're mainly just respiring. Okay. Starting position of the meniscus is here, so all this tube is filled with water kind of to there. All right, the narrow tube contained coloured water. I've just coloured it in to make it sort of more visible. The position of the meniscus was noted at the beginning of the experiment. The tube was left for 20 minutes. The new position of the meniscus was noted and is repeated with another type of seed. So what we would expect is that the meniscus would travel this way, and we would see how far it moved in 20 minutes. Which of the following would be necessary to ensure valid results? Keep the respirometer in the dark. I mean, I would say that's not a bad idea. It's unlikely that the seeds are going to photosynthesize, but if you kept them in the dark, They definitely wouldn't. So let's think about that. Keeping the respirometer at the same temperature. Yes, that's also true. Using the same dry mass. I would say all of them. So 1, 2, and 3. Oh, I got it wrong. I just looked at the exam board and it says only 2 and 3. Okay, so yeah. Sometimes you get these wrong. The answer is C. I think what the exam board is saying is seeds aren't going to photosynthesize anyway because they're seeds. The photosynthetic tissue of the plant is only the leaves, so seeds won't photosynthesize at all. I should have known that. So yeah, the answer is C. You don't need to keep them in the dark because seeds don't have leaves. Presumably the cells aren't differentiated, they don't have chlorophyll, so the answer is C. 12. The student investigated 2 types of seed, pea and sunflower. Pea seeds store mainly starch, sunflower seeds store mainly lipid. Okay, so I'm already thinking this is probably about respiratory substrates. Meniscus would move to the left. Okay, this is definitely not true because if it's moving to the left, we're producing gas. But remember this stuff over here, soda lime, absorbs CO2. So the meniscus is definitely not going to move to the left. It's always going to move to the right. So A is not true. That's not true either because it's again left. Meniscus would move to the right with the pea seeds and further to the right with sunflower seeds. Meniscus would not move. No, that's not true. Okay, well C is correct by process of elimination. If you really want to know why the sunflower seeds move further to the right, it is because if you have a lot of lipid, the seeds need to absorb more oxygen to respire lipid because lipid doesn't have much oxygen in it. It's a lot of hydrogen in the lipid and not a lot of oxygen compared to the carbohydrate starch. It's to do with the different values for respiratory substrates. Whew, these are challenging. So 12 is C. 13. Which of the following statements, A to D, describes and explains the relative yield of ATP in anaerobic and aerobic respiration? Okay, relative yield. Anaerobic and aerobic. Okay, aerobic respiration produces less ATP per molecule of glucose because lactate is converted. Okay, this is one of these questions. Describes and explains. So it has to describe and explain it. Okay, this is correct. Describing produces less ATP. But is it explaining? Because lactate is converted to pyruvate. I don't think that's really the answer. That's not really the explanation. Because, so I'm going to say no for the explaining. Less ATP. Describe, good, yes. Because NAD is not regenerated in oxidative phosphorylation. No, that's not describing either. NAD is regenerated. It's not regenerated, well actually that might be it. Oxidative phosphorylation is sort of to do with it, but let's keep going. I'm a bit confused about that one. Anaerobic respiration produces more that's definitely wrong. Straight out. More ATP, straight out. Okay, then it's B. Sometimes it's difficult to be sure, but if you eliminate the ones that are wrong, it must be B. So yeah, that does explain it. Because NAD is not regenerated in oxidative phosphorylation, I mean, it's worded really weirdly because it's not really about the NAD regeneration. It's because oxidative phosphorylation, doesn't produce extra ATP. Yeah, so the fact that oxidative phosphorylation is written there is the clue you need. Okay, moving on. 2 more multiple choice to go. Ooh, synapses. Which of the following statements, A to D, describes events occurring at a synapse? Okay. Acetylcholine is broken down by enzymes so that it can bind to structure K. There's a wrong and a right in there. This is classic stuff. It's false, even though this is true. Yeah, it is broken down by enzymes, but not so that it can bind to structure K. It's broken down by enzymes so that it can unbind from structure K. B, an action potential causes structure J, where's J, to close. No. Okay, it causes it to open. Structure J is an ion channel. We should label these ion channel. L is a vesicle containing neurotransmitter, and K is your Na+ ion channel / receptor. It's the nicotinic acetylcholine receptor. This is a calcium ion channel. Oh, there we go. C. Yep, that's right. And L. This is almost right, but it's not. Structure L is released by exocytosis. Not really. It's not the structure. It's not the structure L that is released. It's the contents. So the answer is 14C. The neurotransmitter is released via exocytosis, not the vesicle. You don't release a vesicle. The vesicle merges and releases the stuff. All right, GABA is a neurotransmitter. It is. It's involved in IPSPs. Hopefully you remember that. Inhibitory postsynaptic potential. This is when you're less likely to fire an action potential. So it explains it here. It reduces the number of action potentials in the postsynaptic neuron by opening chloride ion channels in the postsynaptic membrane. Okay, so what would that do? It wouldn't bind structure K. It would have its own separate ion channel that it opens, the chlorine ion channel, so that's not right. GABA causes hyperpolarization. Yes, it would make the membrane potential of the postsynaptic neuron go more negative, so sort of beyond minus 70, I guess, so maybe up to minus 80. Definitely doesn't cause depolarization, and definitely doesn't do that, so it's definitely B. Okay, that is the multiple choice, and now we're going to get into some even more challenging long answer questions, and we're really going to be focusing here about technique. Okay, question 16, here we go. All right, so this is ATP. You do need to know this. This is just first question is a knowledge thing. This is adenine. This is ribose. By the way, if it was deoxyribose, it'd be missing, I think, that one. And this is 3 phosphates. So it's adenine. That's the answer. Name the type of reaction that occurs when ATP is converted into ADP. So this would be losing one phosphate. And this, if we're losing something, if we're cutting something off there, it would be hydrolysis. Now, I think in the mark scheme it also accepts dephosphorylation. I don't really use that term very often, but yeah, if you're removing a phosphate, you could call it dephosphorylation. Okay, this next one is a bit more tricky. Part 3. So a teacher told his students that the human body makes the equivalent of its own mass in ATP every day. So yeah, I'm around about 70, 80 kilos. So am I making 70, 80 kilos of ATP every day? Yes, I am. But how come I'm not fully ATP? I'm not just like a big blob of ATP sitting here. Why is that? Well, it's actually only a few grams of ATP in your body at any second. So what's going on? It is constantly recycled. I'll draw a picture and then I'll show you the mark scheme. So if we kind of think about it throughout the day, ATP is broken down into ADP plus PI, and then it's remade. So this idea of constant recycling and regenerating, and this energy is released. And why? Well, it's used to power metabolic processes. And on the other side, respiration of food regenerates ATP. Okay, so that is kind of overview, and now let's look at the mark scheme for those couple questions. So adenine, hydrolysis, and here are the marks for that third, slightly more difficult question. So because ATP is broken down or hydrolyzed, this idea of constant recycling, I think, is what I was getting at. And then it's used to provide energy for metabolic reactions. You have to say metabolic reactions or processes. I would have got 2 there. You could have also said it's not stored or it's used immediately. So yeah, you didn't get. Some people sort of, when I've marked this question in the past, have said used for respiration, and some people did get this a bit backwards. So you might have said that ATP is broken down in respiration, okay? So you need to make sure that you understand this diagram. ATP is broken down to release energy, to power metabolic processes, and we create it in respiration. So don't get those the wrong way around. Right, let's move on. Okay, right, now this question was not brilliantly done when I've looked at some past papers. And the first thing you've got to do in this question is you've got to look at the axes of the graphs and try and figure out what is going on. So, a student investigated the uptake of two different substances, uptake of substances in cells. And the rate of uptake was measured at different concentrations. Okay, so this axis down here, the x-axis, is the concentration. Scientists varied the concentration and measured how the rate of uptake depends on the concentration. A lot of people thought of this as time. They just looked at the x-axis and they thought time and rate of uptake, but they didn't look at that concentration there. So in this experiment, as you increase the concentration, the rate of uptake goes up. So this is the concentration, but we could also view it as the concentration gradient. So as the concentration gradient is going up, the rate of uptake is going up. And over here, as the concentration gradient is going up, the rate of uptake goes up, but then it stops going up. It stops going up, okay? A lot of people, when they do this, they would say things like the rate of uptake rises and then stops, okay? It hasn't done that. The rate of uptake rises and then it levels off or plateaus or reaches a maximum. So people were talking about this part of the graph as if there was no uptake going on. There is uptake going on. It's a high rate of uptake, but it's just reached a maximum rate of uptake, okay? That is really important. So in this one, the rate of uptake keeps increasing as the concentration increases. This screams out passive diffusion, okay? Whereas in this one, something, even if you keep adding more substance, it can't take in any more. So it's not passive, it's not just going through the membrane. Something is limiting. And the thing that is limiting is a carrier protein. Okay, so it could be a protein pump if you're thinking active transport, or a carrier protein if you're thinking that it might be facilitated diffusion. So this one could be active transport, or it could be facilitated diffusion. So here's the actual question. I forgot to even read the question, but if you've done that analysis, when you get to this, you'd be pretty happy. The student concluded that one substance entered the cells by simple diffusion and the other by active transport. And you basically say, yeah, that makes sense. But you say that substance A is diffusion because as the concentration gradient increases, so does the rate of uptake. But that B is probably active transport because as the concentration gradient increases, the rate of uptake increases up to a maximum, at which point it doesn't increase anymore. And that's because all the carrier proteins are kind of being used for transport, and there can be no further increase. So let's look at the mark scheme. So I didn't say this straight line. I think I would have got this mark though here. If I had written it, I would have said as concentration increases, rate also increases. So that is allowed. Graph is a straight line or is directly proportional is also allowed. So therefore it's diffusion. So substance A is diffusion. And then when we get down to substance B, you need to say this: reaches a plateau, levels off. A lot of people were saying this. Do not allow rate slows. Now, would I have? I think I would have. If I'd written it, I think I would have said levels off and stops increasing, or reaches a maximum and stops increasing. I think reaches a maximum would also be accepted, I think, instead of plateau. So yeah, active transport. Okay, so pause if you want to have a more look at that mark scheme, and we're going to move on. Okay, so this second part of the question follows on from the first part. So this says the student added some stuff. Okay, 2,4-dinitrophenol. Some of you actually in my class remembered the DNP, how it affects respiration. You didn't need to actually for this question. You can just take the information they give you. It stops respiration, okay? DNP, no respiration. So what does that mean? Well, let's just scroll up again. Right, no respiration, diffusion, doesn't care. Diffusion is a passive process, it doesn't need ATP, so diffusion, unaffected. This one, substance B, if there's no respiration, then active transport can't happen. So therefore, you would expect the rate of uptake to get much lower. So let's look at the mark scheme for that. Okay, so substance A, which remember was diffusion, unaffected or no change. And the explanate, you do need to get a mark for the explanation. So it does not require ATP, or you could have said it's a passive process. And the next one, substance B, reduced or even no uptake, either one would be fine, or slower, because active transport requires ATP, or is an active process. Now you might have said that it was facilitated diffusion. B could potentially be facilitated diffusion. You do get the intake of some things via diffusion and via a carrier protein. So if you had said facilitated diffusion, then it would be unaffected, because even though it requires carrier proteins, the carrier proteins don't need energy to operate if you had said facilitated diffusion. Okay, moving on. Ooh, it's hard. This is, okay, so this is basically just know your science, how science works vocabulary. Okay, so if you didn't get this right, or if you found this hard, you just got to know your vocab. So you do have to read this a little bit above, because it's about a colorimeter experiment. It says the student used a colorimeter to measure the concentration of each substance in the liquid surrounding the cells. The colorimeter had an analog display, which is weird. I've never seen one like that. So it's got a needle that kind of goes like this to show you the number of absorbance or whatever, to the nearest 0.1 absorbance unit. After the investigation, the student suggested some improvements. This is some old colorimeter they're using. Draw a line between each of the improvements to the corresponding justification. Okay. Colorimeter with a digital display, 3 decimal places. Okay, so that is talking about resolution. The number of decimal places that your measuring instrument can run to is the resolution. So that one goes straight down to here, resolution. Okay, you need to learn the definition for resolution. It's basically to do with the decimal places of your measuring device. Okay, check the zero value of the colorimeter with purified water before use. Okay, so basically your colorimeter, it might have been just like set wrong. So if it's not truly measuring 0 as 0, because pure water should come up as 0, but if pure water comes up as 0.2, then all your results are going to be off by 0.2. So that would be a systematic error, because every result you did would be off by the same amount. It's not a random error, it's a systematic error. So that's this one, this idea of 0 value. For example, imagine if you measured your height with a tape measure and the end had been cut off. Okay, let's say 20 centimeters at the end of the tape measure had been cut off. So instead of being, I don't know, 1.6 meters, you rolled out a tape measure, you go, oh my God, I've grown, I'm 1.8 meters. Everyone in the class, if they use that same tape measure, would come up with an exactly 20 centimeters bigger height than they should have done. That would be systematically wrong by 20 centimeters. For each concentration, repeat the measurement of the rate of reaction 3 times and calculate a mean. So that repeating and making an average gives you a more accurate average, and that reduces random error. Every time you do an experiment, there's a teeny bit of error, a little bit too high, a little bit too low, but if you do many, many repeats and take the average, all those random errors, they average out, because some random errors are a bit higher, some random errors are a bit lower, and then they average out to get an accurate value. This one, this is a toughie. Our students in several schools, okay? This is reproducibility. So repeatability is if one scientist or one group of scientists do the same experiment many times. So, okay, well, let's say one scientist. If you do the investigation and you do it 10 times and you get the same results, it's repeatable because you did it again. But if you do it and then your friend does it and then your friend in another school does it and then someone else in another school does it, that's reproducible. So again, it's definitions. Repeatable, you. Reproducibility, somebody else. Okay? So let's join those up. That one goes to there. And we're done. Okay? Moving on. Okay, so here's question 17. Let's have a look at this. Now, I had to record this a second time because I made a mistake first time I did it. And the first time I did it, the mistake that I did was I just looked down this table here and I thought they were in order. But actually, they're not. That's annoying, isn't it? This one, 0.9, is the highest RF. So that means that that molecule has got furthest. Okay, it travels 90% of the distance of the solvent. So that's carotene, yellow, orange. The next one, the next furthest is this grey one. Okay, so the grey one here, pheophytin, has a 0.65 RF. So that's going to be this one. 3. What's the next one? We've done the top one. We've done the next one. The next one is going to be 0.53. So that's going to be a bluish greenish one. That one. And then after that, so after 0.53, we've got 0.49, which is chlorophyll B, so that's just a green one. And then the xanthophyll one is another yellow. Okay, this would be a bit yellowy orange. Okay, so then it says calculate the RF value for spot 3 and use this to identify. Well, I kind of just worked it out by the order. But you can actually calculate. You can measure this distance here. So you measure that with your ruler. And let's call that the distance S for the spot. I probably should have called it something different, shouldn't it? And then we measure this distance here from the origin to the solvent front. And we'll call that F for solvent front. So the RF is equal to the spot distance divided by the solvent front distance. And hopefully you get an answer which is pretty close to that one. So it's going to be like 0.52 or 0.53, and then you'd identify it as chlorophyll A. I'll show you the mark scheme in a second so you can see the tolerances that they give you. Predict the colour of spot 4, grey. Okay, so I did that by working out the order. So it's going to be grey. Okay, this next one threw a few people. The solvent used for the separation was non-polar. Non-polar. So identify the spot corresponding to the least polar pigment. So if the pigment is also very non-polar, if it is the least polar, if the pigment is non-polar, it likes the solvent. Things that are non-polar get along. So if it's a non-polar pigment, it loves the solvent, it wants to stay hanging out in the solvent. Therefore, it moves with the solvent way up the chromatography paper, and therefore, it is carotene. Okay, so carotene is the answer. And you don't just say it likes the solvent, because it is very soluble in the solvent. Okay, soluble or highly soluble in the solvent. There's my short answer, but if I was writing it in full, I'd write something like we would find in the mark scheme, which is right here. Skip that one, we did that one already. Okay, so we were just looking at this question, spot 5, because it's most soluble in the mobile phase or most soluble in the solvent. Would I get the mark if I just said it's very soluble in the solvent? I don't know, I hope so, maybe not. Most soluble in the solvent, great. And going back to that question here, 0.53 or 0.52 is allowed. Now, correct use of RF value does get a mark. So you could have got a mark if you did this and you had your numbers there, but perhaps you may have just mismeasured somehow. You could have still got a mark. Okay, let's move on. Okay. All right, this one, 17b. Here's the method for the investigation. You read through all the steps, you read through them carefully, you try and figure out what each part is about. Explain why the method included the following precautions. So let's try and find those precautions. I won't read all this out. I think that'll take a little bit long and you've read it already. Explain why the method. Hold the TLC plate carefully by the edges. So where is that? Hold the TLC plate carefully by the edges and avoid damaging. So it tells you right here, avoid damaging the surface of the plate. And the next one, make sure the plate does not touch the jar. Here we go. Okay. Well, the answers here are relatively simple. First of all, if you damage the plate, then you might affect the movement of the pigment up the plate. I don't know if hopefully if you've done this actual practical, the plate is actually it's like a plasticky plate, and then you have this layer of, I think it's silica dust or something on the plate, which has kind of been squished onto it, and it really easily flakes off. So that if you damage the plate, you affect the movement of the pigments. That's the answer. Now, if the plate touches the side of the jar, this is a little bit weird. So imagine here's the piece of paper, and it's leaning against the side of the jar. Okay, there's your jar, and you've got your solvent in there or something. Now, there might be little beads of condensation of the solvent on the edge of the jar. So if the solvent from the edge of the jar gets onto the plate, it will start moving this way. So maybe a spot, which hopefully you put up here, the pigments are moving up, but then suddenly there's solvent coming this way. So then like the pigments are traveling up and they kind of go, whoa, and they kind of get swerved by this solvent that's moving across. So those are your answers, okay? It's a pretty weird one, that second one. But that's what the mark scheme says, and I'll show it to you in just a minute when we've looked at this one. Step 1, why work as quickly as possible? Well, there's two reasons you can go for. First of all, we're using a solvent called propanone in step 1, and it basically evaporates super easily. So if you don't work quickly, then all your propanone may evaporate. Second of all, whenever you're working with a biological material, there's a risk of it breaking down or degrading, and chlorophyll pigment is like that, or the pigments in chloroplasts, are like that. The pigment could degrade or break down, which would be bad for your investigation. So here's the mark scheme. Just have a little look. Pause if you need to have a little look and see how you did. So we're looking at 17b, 1 and 2. Okay, have you looked at that? Let's move on and talk about 17c. Okay, 17c. So you'll see here that I've dropped in a little picture, because this question is really all about the Calvin cycle. And you need to kind of have this image in your mind to be able to do this question and answer it properly. But let's just go back a second. So algae are single-celled photosynthetic organisms, and they did an investigation into the effect of CO2 on the concentrations of ribulose bisphosphate and also glycerate 3-phosphate present in the algae. So first of all, the algae were grown in a solution of lots of CO2, and then it was reduced. And then we looked at the concentration of RUBP and GP for 2 minutes before and then 3 minutes after the reduction in CO2. So the first thing it might be helpful to do, I'll use some different colours, is to highlight RUBP here, and then also look at its position in the cycle. It's here. And then the other thing that's useful to do is to highlight GP here, and also look at its position in the cycle. GP. Sorry, this is an annoying cycle. I just realized I picked this one. This is American labeling, but GP is that, okay? GP. This is GP. There's different names for these things, and I think PGA is, I think what they call it in the States, but we call it GP. And also, whilst I'm at it, I got this image from Khan Academy. Whilst we're at it, let's also look at this one down here, because this we call triose. phosphate, okay? Yeah. So, what's going on? Oh, and the other thing which may be worth looking at as well is just, we'll also highlight CO2, okay, because CO2 is there. So what happens when we reduce the CO2 concentration? For the question, first of all, it's worth 3 marks, high value question. And the question asks you to do 2 different things. It asks you to describe and to explain. So even if you can't explain, you can still get 2 marks for describing. How good is that? You can get 2 marks just for talking about how these highlighted lines move. What happens to the blue one? It goes down. GP decreases. That's worth a mark. What happens to the yellow one, RUBP? Well, it goes up and then it goes down. That's worth a mark. So GP goes down, RUBP goes up, then it goes down. We've already got 2 marks. What about an explanation? Well, the explanation for GP is if we take away the CO2, look, there, then no CO2 can react with ribulose bisphosphate, or RUBP, so this process can't happen, and therefore we can't make any GP from the RUBP combining with carbon dioxide, or carbon fixation. So that's one explanation. The second explanation is a little bit more complex. So for RUBP, initially, if we stop this process, the concentration of RUBP will build up and become quite high because it's no longer being converted into GP. But eventually, if we aren't making any GP, then the regeneration of RUBP is also going to be affected, which would mean that its concentration would fall. Okay, so here's the mark scheme for you to look at. There it is. Pause and have a look if you need to, and then we're going to move on. Okay, question 18. Fura2 is a fluorescent dye that can be used to measure, I feel like this will be important later, measure the concentration of calcium 2+ ions inside cells. So, it says use the information in the figure to explain why Fura2 is unable to cross cell membranes. I think people did this pretty well actually, but whenever you get this type of language in the paper, use the information in the figure, that means you've got to talk about something that you can actually see in the diagram. So what can I see in the diagram? I'm just looking at it. Well, it's big. That's one thing. What else is there? Well, there's a couple kind of benzene rings if you do chemistry. And there are also lots of Cu groups. Okay, these are carboxylic acid groups. So this should hopefully jump out. We've got a minus charge. We've got a minus charge, a minus charge, a minus charge, and a minus charge. This is really charged. Okay, so there's 2 ways you can approach this question, and there's 2 different ways of getting 4 marks. It can't cross membranes, one, because it's a large molecule and it's too big to get through the phospholipid bilayer, or you could say it is a charged molecule, and charged molecules can't pass the phospholipid bilayer. Notice that both times I said phospholipid bilayer. You have to say that, otherwise you didn't get the marks. So here's the mark scheme. Have a look. In both cases, you could say it's charged or polar, and it won't fit through the phospholipid bilayer, or you can say it's too large, it cannot pass through the phospholipid bilayer. So you have to say bilayer. Look at this over here, it's harsh. Ignore cell membrane. Ignore cell membrane. Isn't that mean? Yes, I think it is quite mean, but you've got to say bilayer because it's A level. Cell membrane is kind of GCC. Phospholipid bilayer is what it is. All right, moving on. Scientists have used Fura2 to study the role of calcium ions in the synapses of living nerve tissue. Okay, because remember, it can be used to measure the concentration of calcium ions. Fura2 was injected into a single sensory neuron. Maybe that's relevant. I'm going to highlight that. Was connected by a synapse to a relay neuron. Okay, right. Fura2 inside of the neuron was observed using a confocal microscope. Explain one advantage of using a confocal microscope in this study. Here's a GIF of a confocal microscope study. Here you go. And this is great because we can actually see how living tissue is working. So that's the main answer, is that we can use living tissue to actually see processes, okay? Because if something is dead, you can't see a process. You can't see an action potential in something that's dead. So living tissue is very helpful. Also, you could have said something about that confocal microscopes can kind of take an optical plane through a large object to look at what's going on inside. The mark scheme I'll show you just a minute once I've looked at this second part of this question. So B part 2. This is about looking at the data and drawing conclusions. So let's analyze it. The sensory neuron was stimulated electrically and the Furrow-2 fluorescence in the synaptic bulb was measured. So remember the synaptic bulb, I'll just sketch it a little bit, is, tech problems, is looking a bit like this, okay? So this is the synaptic bulb here, the presynaptic neuron, and this is the postsynaptic neuron down here. So this is the postsynaptic neuron, and this is the synaptic bulb. So if we have low electricity, we don't have any fluorescence in here, or low, and over here we've got minus 60. So this basically means low electricity, we could also just write low calcium here, okay? Low Ca2 plus, and that is resting potential, minus 60. There's no action potential going on, that's resting potential. What about medium? With medium electrical stimulation up here, we do have medium calcium ions, because remember, Furrow-2 measures the calcium ions. So if we have calcium ions going in to here a little, then we do get an action potential in the postsynaptic neuron. That plus 40 is an action potential. And if we have high calcium, well, high electricity, high calcium, then we do get an action potential. Okay, so there's kind of two things sort of going on here. First of all, that as we increase the electrical stimulation, we get a direct result that we also increase the calcium. So those two are positively correlated. Increased electrical stimulation, increased calcium ion intake, or increased calcium ion presence in the synaptic bulb. And the second thing is that having low calcium is not sufficient to trigger action potential in the postsynaptic neuron, but having medium or high calcium is sufficient to trigger an action potential in the postsynaptic neuron. So let's look at the conclusions. The scientists concluded that changes in the concentration of calcium ions in the presynaptic neuron caused an action potential in the postsynaptic neuron. So, I mean, it's so tempting to just say, yeah, I completely agree. But that's never what you need to do. Whenever it's evaluate a conclusion, if you see those two words, evaluate and conclusion, you've got to say some positive and some negative stuff about it, okay? Now, generally speaking, it can be 50-50. In this case, maybe I think it's 75-25. And let's look at the mark scheme to see what they want you to say, okay? So we talked about this factor. The fact that the concentration of calcium is proportional to the strength of stimulus. That is a mark point for the conclusion. Here, we also talked about this. We talked about medium and high causing an action potential. And then that's sort of describing the conclusion. Let me just go back to the question for a second, sorry. In the question, it says evaluate and, I almost didn't do this, and explain. So evaluate the conclusion and then explain the conclusion. So if we look at the mark scheme there, these are kind of evaluate parts, and this is explain. Okay, so we evaluate and explain. So this is evaluate, this is explain. And then this is again explaining, talking about the release of the neurotransmitter, the named neurotransmitter. It could be acetylcholine you could get, ACH for short, acetylcholine will give you the mark. And the fact that this causes the sodium ion channels in the postsynaptic neuron to open. And then if the threshold potential is exceeded, then we get an action potential. So remember, threshold potential is about minus 50. If we don't go past that, then you won't get an action potential. Now, the thing about the negative thing that you could say is the kind of the electrical stimulation going from medium to high, there's no difference. Okay, so this bit, calcium changes from medium to high, but no change because it's an all or nothing event. So that is the answer. Moving on. Oops, sorry, I'll just go back. If you need to pause, look at this mark scheme, pause there, and we shall now move on. Okay, so here is question 19. So here we've got mitochondria. So when we're looking at mitochondria, we would never see this detail with a light microscope. So this is definitely an electron microscope picture. State the type of microscope. Well, electron microscope's not going to cut it. Is it scanning or transmission? And it's transmission. Transmission, electron microscope. I'll write that out in full, but I'm just being just noting it for you. So TEM, or transmission electron microscope. And that is because it's a two-dimensional image, not 3D. So what are the structures here? I mean, it's a bit difficult. You really need to kind of zoom in and look closely. But it would look like M is over here, is in the kind of solution, and N is this kind of line. So make sure you're looking at the correct thing. So N is there and M is there. So M being the kind of solution is the matrix. And N being the membrane is the crista or cristae. Zoom out a little bit. Now, it says, describe two ways in which the structure of mitochondrial membranes is related to the function. Okay, is related to the function. And now, it's so tempting to say high surface area here, but I mean, I probably would have said that. I've had a look at the mark scheme before, and it doesn't accept that. But so I think you need to go into a bit more detail. I think when you're answering something like this, you kind of almost want to cover yourself. So you say the cristae have a high surface area to contain many electron transport chain proteins. Okay, so that's kind of covering you in case it's surface area they're looking for, or if it's electron transport chain. So they have a high surface area to contain lots of electron transport chain proteins, or you could say a high surface area for lots of ATP synthase molecules. And let's look at the mark scheme. I'm pretty sure that's what they want. Contain electron carriers or ATP synthase. I might have only got one mark out of that one, so this one I don't think I would have got. So it's the site for chemiosmosis or ATP synthesis. Or you could have talked about formation of proton-ion gradients, or the fact that the outer membrane, I doubt you would have got this, but yeah, the outer membrane of a mitochondria is permeable. It's the inner membrane that is impermeable to H+, but the outer membrane is pretty permeable, so that things like pyruvate, etc., can move into the mitochondria. Pause there if you want to look, and we'll move on to the next question. Okay, C. The endosymbiotic theory suggests that mitochondria may have evolved from bacteria that were taken inside other cells. How did they get once they got sort of swallowed up, then eventually the cells evolved into eukaryotes. So there are many features that support this theory. I'll show you the mark scheme in a second, but here's just a list. So double membrane. Mitochondria have their own DNA. They have small ribosomes that are very similar to bacteria. Those are 3. There's a few more. Okay, so on to part 2. Explain why early eukaryotes were able to grow more quickly than cells that did not possess the mitochondria. Well, there was a massive advantage to having mitochondria, and that advantage was that you've got aerobic respiration happening, which means more ATP, which means you can do more things. So you can carry out cellular processes or metabolic processes a lot faster. Let's look at the mark scheme for both of those questions. So the mark scheme for the points that would suggest endosymbiotic theory is right here. And the one that we've just been talking about is down here. So we'd be able to respire aerobically, more ATP, and ATP is needed for, I think you need to say metabolic processes. Yeah, this is down here, this point here, for more metabolic processes. So have a pause there if you want to look at the mark scheme for either C1 or C2. Okay, let's move on. To 20. Okay, question 20. This is an experimental method, so the first thing to do whenever you're doing this sort of thing is reading through the full experimental method. So you've got a stock solution of yeast, it's got 10 grams of yeast, and it's got pH 5 buffer. So we already know it's the same pH because it's a buffered pH. Now we've got solutions of each sugar that we're going to test. 5 grams of sugar in 250 centimeters of distilled water. Keep the yeast and sugar solutions in a water bath at 35 C until required. So that so they equilibrate in temperature, so everything's at 35 C. Set up the apparatus with a 250 centimeter conical flask with a rubber bung to a gas syringe. Add the yeast solution, or 25 centimeters of the yeast solution, and 25 centimeters of the sugar solution to the flask. Remember, these have been pre-warmed. Immediately connect the flask to the gas syringe and start the clock. Record the volume of gas produced after 30 seconds and then every 30 seconds for 7 minutes. So timing for 7 minutes. Repeat the experiment 5 times for each different sugar. Prepare a fresh yeast solution for each set of sugars. All right, these last 2 steps are the ones which you kind of seem a bit suspect, I guess. It could go wrong. So it just says, describe 2 precautions the students should take between each experiment. And it does give you that clue. It says between each experiment. Okay, so after the first one, what should you make sure to do to ensure repeatable results? And I think most people got the first one, which is just to make sure that you rinse out the conical flask. If we only got one conical flask, make sure we rinse it out clearly. And the next thing is prepare a fresh yeast solution, is to make sure that each yeast solution is prepared at the same concentration. Is that right? We'll look at the mark scheme in a second. I can't quite remember the second point, to be honest. 20A part 2, the student's procedure did not include a negative control. Describe one negative control that the student could have used. Well, a negative control is something that you would expect not to work. And basically, if it does work, if you do a negative control and then you do get respiration, then something weird's going on. So what would you expect to not respire? And in this one, the answer they want you to say is dead yeast. So you have to say boiled yeast. Okay, I can see how some people might have just put water, just water and sugar. But I think they actually want you to say dead yeast. Let's look at the mark scheme. They don't want you to say dead yeast, they want you to say boiled yeast. Okay, so here are the mark scheme. So yeah, you've got a few things actually here. Oh yeah, I forgot loads. So rinse, change flask equipment, stir the yeast stock solution. Oh, that's an obvious one, isn't it? Because if you've got the yeast stock solution in a big old 250 centimeter flask like that, you stir it up and all the yeast is in here, but then it will settle down to the bottom. So you've got really lots of dense yeast at the bottom and not so much at the top. And if you take from different points in the flask, that's going to affect how many yeast cells there are. So that's a huge error there. Same type of yeast. Yeah, I think I think I mentioned that maybe. That's the obvious one. This is a good one. Ensure the connection to the gas syringe is tight. Because each time, if there's a tiny leak, then you won't get any gas in the syringe and it will look like the yeast is not respiring. Also check the temperature of the water bath. Yes, that's a really good point. Maybe it's got too cold. You never know. So those are all good points. Boiled and cooled yeast use buffer instead of yeast. So that's kind of, I would have said just use water instead of yeast, but use the buffer instead of yeast. Okay, moving on. Aha. Okay, describe how the student could use the graph to calculate the rate of respiration for each type of sugar. Mean values for each sugar and plot the results in the graph. Okay, so how do you calculate the rate? Well, first of all, rate is volume over time. Okay, so that's the first thing. Rate is volume over time. Now, where to measure the volume and time, that's a bit tricky because it looks like that most of the glucose has kind of been respired by here and then it kind of there's not much more gas produced, probably because there's not much glucose left. So that's a bit tricky. So I would suggest that we would probably draw tangents on these graphs around about when the sugar seems to be kind of being respired with a bit of consistency, should we say? So I would say at this point we draw a tangent there. And then at this one here we draw a tangent sort of there. And this one here, maybe we draw a tangent kind of there, I guess. Yeah, there, okay? So we look at the rate of each tangent. So if we draw the tangents, and then using the tangent, we take a time and a volume of gas, and we'd work out the gradient of each tangent, like so. And that would give us the rate, okay? So that's how we would do it. What do we need to say when describing that process to get the marks? Let's go to the mark scheme. So, first of all, this one I wouldn't have got. Rate of respiration is proportional to the rate of gas production. It's important to say that we weren't directly measuring the rate of reproduction. We're measuring gas produced by respiration. Did I say reproduction? We're measuring the rate of gas production, which can be used to work out the measure of respiration. So it's proportional. Tangent, said that. Gradient, said that. Volume of gas divided by time, said that. And I'm not sure, actually, I'm not sure if I entirely agree with number 5. I think we would compare, well, if you were just looking at the total volume produced, we'd look at the same time period between sugars. Maybe does that mean you should work out the gradient over a 1 minute change or something for each sugar? I'm not really sure about that one, to be honest. Okay, so this is a really, really important question. It's a 6-mark question, and we really need to focus on how we can answer these questions with skill. So if you have zoned out, then now's the time to wake up. And if you want to pause here, grab a cup of tea and come back with fresh eyes, then you could do that as well. So, are you ready? Okay. So, on any question that is a 6-mark question, I say any, but loads of them, okay? On loads of 6-mark questions, they want you to do not one thing, but 2 things, or even 3 or 4 things in the question. So the part of the preparation that you must do is work out what are the multiple things that you need to do in the question and do a little plan. So that's what we're going to do here. Let's look at the question. The student concluded the following from the graph. The rates of respiration with glucose, maltose, and sucrose were very similar. Okay, so we'll call that conclusion 1. Conclusion 1, similar, we'll call it. Similar, conclusion 1. And then conclusion 2, the yeast could not hydrolyze disaccharides. I'm going to call that conclusion the disaccharide conclusion. Now, the question says, evaluate the student's conclusion. The next thing you need to know is whenever you get a question like this in A-level biology, evaluate the student's conclusions, they don't want you to say, yeah, the student's right. Or just like, they're really right because of this, this, this. They're going to be partially right. They're always going to be partially right, partially wrong. And so you need to talk about both sides of the coin. So here's what I want us to always do, or you to always do when you're planning it. You need to lay out a grid, okay? So draw a grid. Like so. We've got conclusion 1, the fact that they're similar. And we've got conclusion 2, the fact that disaccharides aren't hydrolyzed, they say. And then we need to talk about things that support the student's conclusion, positive things, pro-conclusion, and things that are against the student conclusion, negative things. Now here's the image, so we can kind of refer to it more easily. Well, the first thing, let's look at the similar conclusion. The rates of respiration, glucose, maltose, and sucrose are very similar. Yeah, look at the gradients of the tangents, okay? They're all very similar. Gradients, similar, okay? They're all pretty similar. And actually, you could work out the gradients for each one and state them, and the fact that they're all very similar. What about, and the other thing is they all reach similar volumes of gas eventually. That you could probably also say that. Eventually, volumes of gas are approximately the same. I don't know if that's worth a mark, but it's worth a go. You can't get marked negatively for saying stuff that's not quite right. Or what about negative? Well, they don't all start at the same rate. Okay, so glucose kicks off pretty fast, whereas there is a delay of a couple minutes, maybe 2 or 3 minutes really, before maltose gets into gear. I'd say about 2.5 minutes to 3 minutes before maltose hits its stride. And for sucrose, potentially even more of a delay. Sucrose is only going at its maximum kind of gradient around 4 or 5 minutes. So we need to talk about the delay there. And we could also talk about, so delay and starting. And we could also talk about when they sort of, when gas stops being produced. So glucose finishes quite soon. So around about 4 to certainly 5 minutes, glucose has already stopped. There's no more gas being collected from glucose. Whereas maltose stops at about 6 minutes and sucrose stops at about 6 minutes. So we can say different end points. I'll just write it as a note, but you probably write it in a bit more clarity. So we've looked at positive and negative of the first conclusion. What about the disaccharide? Can we have a positive and a negative? Remember, he said that the conclusion, he or she said, that the yeast could not hydrolyze the disaccharides. So which ones are disaccharides? I'm just going to highlight them. So let's highlight the disaccharides in blue. So is maltose a disaccharide? Yes, it is. 2 alpha glucoses. Maltose is a disaccharide. Is glucose? No, that's just a monosaccharide. Is sucrose? Yes. Is lactose? Yes. Okay, so actually there's 3 disaccharides there, and 2 of them can be hydrolyzed. So maltose, and worth saying it, 2 alpha glucose, can be hydrolyzed and respired, so that's clearly against their conclusion. So can sucrose, and again, say what it is. It's alpha glucose plus fructose. That can be hydrolyzed, so that's definitely against the conclusion. However, lactose, definitely doesn't get respired. You can add a little bit more to this and maybe say that maltose and sucrose maybe can't be respired immediately, but first need to be broken down or hydrolyzed. That would explain the lag. So some enzyme will need to split the maltose up into its alpha glucoses, and it would do the same thing for sucrose. It would break the glycosidic bond between the 2 sugars. Okay, so that's the way you address the question. And if you cover all 4 quadrants, you're going to give yourself the opportunity to get 6 marks. In an exam like this, if you only talk about 1 quadrant, you're probably going to get 1 or 2 marks. And maybe if you talk about 2 quadrants, you might get 3 or 4 marks. So let's say you did this. Okay, you talked about the top 2 bits. You just talked about the first conclusion. You gave some positive and some negative. Top maximum you're going to get is 4. You need to talk about all the squares of your grid to get 6. Okay, so let's look at the mark scheme, which is here. I'll zoom out just a little. So this is why it's so important, because look at the levels of response that you can get. For level 1 or 2, incomplete evaluation for or against statements for 1 conclusion. Okay, so let's actually just think about it in terms of the grid. So if I've got a grid with 4 squares here, can you remember what we just did? I hope so. What do you need? Okay, so if level 1 and 2, we're talking about for or against statements for 1 conclusion. So we're really talking about you've done 1 square. Level 2, 3 or 4 marks, an evaluation of 1 conclusion for and against, or both, or for or against statements for both conclusions. So you're basically talking about 2 out of the 4 grids here. Could be this way, or it could be this way, if you see what I mean, okay? And 5 or 6, you need to kind of have all, you need to cover each grid square. So if you do that grid method of planning, you're going to stop yourself from just focusing too much on one sort of corner of your grid, and you're going to enable yourself to get 6 marks, okay? So pause and have a look at the mark scheme there, if you need to. And there's a few more below, which I'll just scroll down for. But pause and look there if you want. And there's some more there, okay? So pause and look there as well. All right, I just briefly want to show you some student example work as well for that question. Here's a really, really nice answer, but this student did not cover all the different quadrants or corners, let's say. So they had a plus statement for this conclusion S, similar. They had another plus statement for the S. They calculated the rate, which is really, really excellent. Great maths there. And then they only had one plus statement for the this one, the yeast. So they basically hadn't recognized that maltose and sucrose were also disaccharides, and there were. So there was no against statements. They were very complimentary about the students' conclusions, but they didn't take them apart. Remember, you've always got to, they're partially right. The students' conclusions are always partially right, so you've got to give some negative statements. So if you want to, you can pause and have a read of that. Really well written. However, this student couldn't get 6 marks because they didn't talk about each of the grid squares, and I'm sure they would have got 6 marks if they'd used that grid method. Okay, so on to part C of this question. The student wanted to study the effect of different sugars on the rate of growth of yeast. They used a colorimeter to measure the absorbance of a culture of yeast cells. Why? Well, because if you look at the absorbance, it kind of equals or is proportional to the concentration of yeast cells. But as the yeast cells were multiplying and becoming very sort of concentrated, it was necessary to dilute the sample. So how could you use 1 centimeter pipettes and a 10 centimeter measuring cylinder to dilute the sample so that it was 10,000 times less concentrated? Now it's tricky, but they have given you clues there, okay? So I've put this diagram on the right to show a kind of schematic of dilution plating. So what you would need to do to go from the original concentration start until a 1 in 10,000 dilution here is you need to do 1, 2, 3, 4 times 10 dilutions, okay? So a lot of people, it was very easy to make this mistake, but a lot of people got it slightly wrong. So let's look at how you do 1. So here's I'm going to draw some terrible diagrams, okay? So here's your original yeast culture, okay? And you have your measuring cylinder here. There's your measuring cylinder, okay? Remember, it goes up to 10. Now you take using your pipette, here's a pipette, again, such a bad diagram. You take your pipette and you take out the solution and you put 1 milliliter in there. So you've got 1 milliliter in your measuring cylinder. And then you make it up to 10 with water. So that's one way of saying it. Another way of saying it in terms of mass is you need 9 mils of water. Okay, not 10, but 9. 9 mils of water and 1 mil of the original yeast suspension. Okay, once you've done that, you just drop that in there. You don't just leave it in there. You, maybe with a rubber glove on your thumb or something, you invert it a couple times. You give it a good old mix. You make sure it's properly mixed, and then you've got your new times 10 dilution. Then you can go into this times 10 dilution and repeat the process. Take 1 mil out of it, put it in a new tube, make it up to 10 by adding 9 mils of water, and so on. So the first time you do the dilution, you've got a 1 in 10 dilution. The second time, you've got 1 in 100. Oops. The third time, you've got 1 in 1,000. And the fourth time, you've got 1 in 10,000. Whoa. The pen's gone crazy. That's better. So let's look at the mark scheme for that question now. So the correct description of the 1 in 10 dilution, so you have to have this idea of 1 and then making it up to 10, or 1 plus 9. You need to get the number of dilutions right. That's a total of 4 dilutions, so 1 and then 3 further. You can get marks for giving the correct values of the dilutions, like this is a 1 in 10, and then 1 in 100, 1 in 1,000, 1 in 10,000. And then you can also get a mark for talking about the mixing of the yeast suspension at each stage. Okay, on to this next part of the question. So this is a simple recall question, relatively speaking. Okay, so how would you observe using a microscope and measure the diameter? So 2 things. I bet all of you probably put 1 thing. Eyepiece graticule. That's the first thing. What's the other thing? It's the stage micrometer. Okay, because you've got to use them together. So the eyepiece graticule is basically, it's in the actual eyepiece, and you look down it and you can see the lines on it. But how do you know how big a line is? This, the stage micrometer is basically a microscope slide that has known markings on it. And you use that to calibrate the eyepiece graticule first. Then you know what each line on the eyepiece is that you're looking at, and then you can measure the yeast cells. Simple. The student prepared a starter culture using 2.5 times 10 to the 3 yeast cells in 1 litre of nutrient bath. The average mass of a yeast cell, oh gosh, is not a lot. 2 times 10 to the minus 11. So that is the number. Okay, this is not so bad. So it's just, so if we know the mass, the total mass, and we know how much each yeast cell is, so we could do it. We could write it this way. So number of yeast cells times the mass per cell, mass PC, is equal to the mass. Yeah. So how would we work out the number? Well, we do the mass divided by the mass per cell. That makes sense. So we move that over here, divide it. So number is equal to the mass divided by the mass per cell. So let's do that. So it's the mass, which is 2.5 times 10 to the minus 3, divided by 2.0 times 10 to the minus 11. Now we can do some cancelling here. So the minus 3, get rid of that. And then this would, we take 3 off that, so that would become minus 8. All right, and then we can plug that into a calculator and we should get the right answer, which is 1.25 times 10 to the 8. There we go. So yeah, we get, this would give us 1 mark, the working that I did. And plug it into a calculator and you get this second mark here. Or just 2 marks for the correct answer. Oh, this is a horrendous question. To be honest with you, I'm not even really sure how you do this question. Out of the people who did this in my class, only 1 person got it right. And I'm very impressed. I don't know how you got it right. But there's a little bit you can get right, even if you're not entirely sure. Population of this yeast cell doubles every 90 minutes when growing under ideal conditions. A different starter culture was prepared containing 1 times 10 to the 7 cells. This is super mathsy. Okay, so how would you expect it to go? So log 10, this is basically just testing your understanding of, do you know what log 10 is? So log 10 of 1 times 10 to the 7 cells is 7. You might have guessed that if you just saw the 7s and you thought, well, I'm just going to put it at 7. And if you did, you guessed right. So we start it there. How would it be off every 90 minutes? So we can kind of almost mark in the 90 minutes here, because 90 minutes is an hour and a half. So the 90-minute intervals are like this. So it's going to be, how's it going to go? It's going to go 1 times 10 to the 7. After 90 minutes, it's going to be 2 times 10 to the 7. And then it's going to be at 3, it's going to be 4 times 10 to the 7. over here, and then sorry, so 1 times 10 to the 7, 2 times 10 to the 7, 4 times 10 to the 7, 8 times 10 to the 7, and then it's going to be 16. So over here is going to be 16 times 10 to the 7. Now that we could actually write as 1.6 times 10 to the 8, couldn't we? So that's kind of, we're in the 8 range and we're probably up here somewhere. We keep going, so that's there. We basically just got to brute force it. You keep going, and you work out what it is finally at 15. And it turns out it's about here. So you enter 1 times 10 to the 7. You have to trust me on this. And then you do times 2 once, times 2 again, 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 and again, and again, and again, and again. Okay? So how many times? That's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Goodness. And then you get a number, and you plot it, and you work out how many times 10 to the what it is, and it'll be up here. And it's a straight line. It goes like that. It's really hard, that question. Well done to the person who got it right. Well done, you. So, okay, if you're wondering how many marks do I get for it, it is 1 mark for a straight line, 1 mark for starting at 0.7, and 1 mark for ending at 15.10. Okay? What's that even doing in a biology paper? I'm not even sure. That feels like physics to me, but oh well. Okay, here's question 21. This table, you have to decide whether there's cartilage, elastic fibers, and goblet cells in these different parts of the mammalian ventilation system, the lungs. Now, first thing to do is I would say that basically as we're going, this is basically going deeper into the lungs. And as we're going into deeper lungs, we're getting less and less cartilage. And elastic fibers are pretty much the same all the way through, and then goblet cells are going to be getting less and less. But exactly what level is the key thing you have to know. So here's the answers, just going straight to the mark scheme. So elastic fibers present all the way through there. Goblet cells just in the trachea and the bronchi. And cartilage, you could have said trachea, bronchi. There is some cartilage in the bronchioles. The reason this is either cross or minus is there are different types of bronchiole, really. There's the kind of wider ones, which have cartilage, and then there's the thinner ones, called respiratory bronchioles, that don't have cartilage as they get sort of thinner and thinner and lead to the alveoli. Okay, moving on to 21b. So, ventilation of lungs creates air movements can be measured and recorded. Now, this apparatus here that is used to get this trace is called a spirometer. Easy to confuse it with a respirometer. But they're different. Okay, so here's a little picture of a spirometer just in case you couldn't remember. Here it is. And now it's gone. Okay, so the question asks us, use the trace to estimate the maximum value for tidal volume during the first minute. Okay, so there's some important stuff there during the first minute. So we can only go up to 60 seconds, so we're looking at this time. So where is the tidal volume, the maximum there? So if this is all tidal volume, I would probably say that the maximum is from this point to this point. So that point there is 2.4. Okay, 2.4. First, you need to make sure that you're looking at the scale correctly. So each individual miniature square here is worth 0.1. So that's 2.4. And then there is going to be, I would say, 3.55. So if you do that calculation, 3.55 minus 2.4, you should get your answer. Just doing that now, 3.55 minus 2.4 equals, so I'm getting 1.15 dm cubed. Let's check the mark scheme. Yeah, so 1.1, it says allow in the range from 1.0 to 1.12. So, okay, moving on. After 60 seconds, the subject is told to breathe in as deeply as possible and then breathe out fully. Use the trace to calculate the vital capacity. So the vital capacity is the big capacity. So again, we're going to just take some numbers here. It's from down there up to there. So that is going to be 4.7. So it's 4.7 minus, and that is values 0.2. So 4.7 minus 2 equals 4.5. It's the answer for vital capacity. Again, checking the mark scheme. Yep, 4.5. And 2 marks for that one. So really this is just about knowing your capacities. The tidal volume is just kind of when you're breathing sort of relatively normally without thinking about it. And then we call this volume here is the vital capacity, and it's when you're exhaling as much as possible under conscious control, and then inhaling as much as possible under conscious control. Now, the reason that's important to understand is because it comes up in the next question. So, the next question is another 6 marker, okay? So, remember what we said in the first 6 marker. It's very helpful to first analyze the question and understand what you're being asked to do. And because normally it's not just one thing, it's like 3 or 4 things. So let's look at it. Compare, okay, so that's similarities between. And contrast, that's differences between. The mechanism of expiration during the first 60 seconds versus when they were told to breathe out fully. Okay, all right. Now this is very important. Quite a few people kind of lost this bit here. Okay, the mechanism expiration is breathing out. How do we breathe out? What muscular contractions or not are required for breathing out for expiration? So let's draw a grid. Okay, let's draw the grid. So we've got compare. So that's similarities, let's say, and we've got contrast, that's differences. Similarities and differences. Got to give both. And so that's tidal, first 60 seconds is tidal volume, and the other one is expiration, breathe out fully, so that's, you call that forced expiration. So compare tidal volume and forced expiration, and then sort of down here, it's kind of a grid, but it's got 3 things. So obviously, if there's similarities, what similarities are there between expiring with tidal volume and forced expiration? Well, when we just breathe out without really pushing, the diaphragm relaxes. And what else happens? And then it rises up. And when it rises up, the volume of the thorax decreases, which leads to pressure rising, and which leads to expiration. Okay, pressure rising and then expiration out. Contrast that with differences. Okay, well, there's no muscular contractions during tidal volume expiration. Okay, obviously you need to move some muscles to breathe, but that's actually for inspiration. That movement is a contracting of the muscles in the diaphragm which pull it down. But to breathe out, once you've got a full breath, it's actually a passive process. So that's the key thing, passive. No muscular contractions. And also, when we're doing it passively, the rib cage can just passively fall because it has weight. So your rib cage can just passively fall. However, if you're doing forced expiration, FE, forced expiration, then you contract your intercostal muscles. I believe these are the internal intercostal muscles in your rib cage, but you didn't need to say that. You contract your intercostal muscles, and that can enable you to kind of reduce the volume, let's say it properly, reduce the volume of the thorax even more and expire even more air. Okay, so that is the kind of grid method for planning, and then you'd write this. Okay, let's go straight to the mark scheme and see what you need to do. So, once again, why is the good grid method so key? Well, let's look at the different levels of response. So, level 1 or 2, a description of normal expiration or forced expiration. So you're kind of not really comparing and contrasting. You're not really listening to what the question is asking you to do, which is compare and contrast. You're just saying what you know about one thing, and not even both things. You're just kind of defaulting, oh, I know how we expire, and you're just saying that. Only 1 or 2 marks available for that. Level 2, 3 or 4, a sound description, so detailed, of normal expiration as passive, and then some comparison. So you've kind of described well one of the processes, and you've started to kind of really address the question, which was the compare and contrast. But then for level 5 and 6 at the top here, you need to really answer the question, a good description of normal expiration, and a good contrast with forced expiration as an active process. Okay, you have to really go for the command word of the question, which is compare and contrast. So the scientific points are over here, which we've kind of talked about. This is normal expiration or tidal volume. And then this down here is the forced expiration, which is about when you're really pushing out. Okay, so if you want to pause the video there, look at what points you did say. If you're in my class and I've marked your paper, the way I have done so is I think I've kind of done stuff like this, sort of like N plus or like N tick for like normal, points to do with normal. And I think I've done F for forced and tick for points to do with forced. So look out for that notation on your paper. Pause the video here, have a look at your answer. Oh, and it's the last question. Okay, great, last question. So this is just a nice, easy 5 marks at the end. You know, what a nice little bonus at the end. Hopefully you got some good marks in this. So large organisms have a large surface area. Okay, think about, you know, an elephant has a big surface area. So SA for that one. But they have a small surface area to volume ratio. Okay, I'm just going to write shorthand, SA to V. Because the bigger an object is, the smaller the surface area to volume ratio is. You may remember that you've done calculations involving cubes of different sizes. That's supposed to be a cube, small cube, big SA to V ratio. If you then increase the size of the cube and do the same calculation, it has a much lower, even though it's exactly the same shape and kind of relative dimensions. A specialized exchange surface and a circulatory system, so CS, circulatory system, to move materials from the exchange surface around the body. And features of an efficient exchange surface are a good blood supply maintains the concentration gradient. Otherwise, the concentration gradient would sort of dissipate as things diffused across whatever membrane it is. And a short diffusion pathway or diffusion distance. That's this one, dP. Okay. Okay, we've reached the end of that run-through video. So if you need to go back and look at any other questions, please do. And I want to see, if you're in my class, that paper annotated absolutely fully with all the correct answers written on, notes in the margin, places you went wrong, and we'll use that in class to work on the areas that you need to work on. All right, see you next time. Bye-bye.